So today it's my great pleasure to introduce how statistics may play a very useful role in genomics. So I title my lab as the junction of statistics and biology because we try to build a bridge to really make statistics something useful in data analysis and make our analysis results more reliable. So I will begin my talk by showing this picture I saw on social media after the Nobel Prize of last year in physics. So this is a very interesting figure to me because it tried to characterize physics in two dimensions, the object we study and the method we use. So in either dimension, we can go from being very fundamental to being very liberal. That means we can be more open-minded, more relaxed in the objects we study, right? From physical objects to anything related to reality, like neural networks. And the method we use can go from physical laws to also computer algorithms. So neural networks seems to belong in the most liberal side for both dimensions. So this figure made me wonder about one thing. That is, for the field I study, genomics, is it a liberal discipline? And I think my answer is yes, for several reasons. First of all, genomics is a very interdisciplinary nature. So as we have seen from the previous three talks, we can see that many techniques from traditional fields were used to study the genomics, right, in our cells, using genomics as technology to study molecular biology at a massive scale in our cells. And also because we often generate data before having hypotheses. So genomics is a very data-driven field. So we have the data, and given the same data, we can come up with many possible hypotheses that we can study. And third, because of data-driven focus, there is a very rapid evolution of computational method. For example, for single-cell data, just one example, there's a website that tracks the number of single-cell tools we develop, and this number has been growing. So given so many methods, how do we choose them for our data analysis? As a result, we can have flexible analytical approaches. That means for different labs analyzing their in-house data, they may have their own pipeline, right? So for all those reasons, I will call genomics a liberal discipline. But what is statistics, my other part of my lab? So I try to reflect the meaning of statistics. In particular, nowadays, I know that many people, if you heard the term data science, you may think more about computer science, because I often had the conversation with people when so I have some data science label with my title or with some award I got, they will say, oh, you must be a computer scientist. And they will never say, oh, you must be a statistician. And I wonder why. I think there are several reasons behind that. Maybe statistics has been, during its own evolution, too concentrated on mathematical proofs. And so gradually detached from real world data analysis. That is one reason. But despite that, if I trace back to the history of statistics like 100 years ago, why did we ever have this discipline? I think it's trying to ensure rigor in data analysis. Let me just try to use this famous example, two-sample t-test, to remind you what statistics was about at that time. And I was very fortunate to be able to visit the Guinness Brewery in Dublin, Ireland. And I saw a label of this guy, Willem Gosset. And he is actually the developer of the t-statistic for this two-sample t-test. But when he published his work, he used the pseudonym student to just be modest. So because of that, the test was referred to student t-test. And the problem was very real. And he worked in this brewery, and they need to decide whether they should change to a new way to brew their beer. So basically, they have a traditional way to brew the beer, and they have a batch of beer brewed in this way. Later, they invented a new way to brew the beer, and they have a batch brewed in the new way. So the question is, if we have like a metric to measure the quality of beer, can we actually say reliably that the new way gave us better quality beer than the old way? That's a question. So the problem can be abstracted as comparing two sets of numbers, right? The metrics in the old way and the metrics in the new way. So we know that if we don't know any statistics, we know that we can just compare the averages of the two batches, see which average is higher. But we know that the two numbers will never be perfectly equal. Then how large is the difference large enough to be meaningful? The two-sample t-test was designed to answer this question. So essentially, it tried to account that the fact that even within the same batch, not every beer has the same quality measure. Then how do we account for such uncertainty in our decision when we compare the two averages of the two batches? So it's about rigor. So I will talk about two examples in this open science day talk. I'll try to convince you that rigor is something we should bear in mind, and data analysis is not as straightforward as we might think. So the first example is regarding this commonly performed analysis called differential expression analysis. So RNA sequencing is the technology that people mostly use to perform this analysis, try to identify some interesting genes for their downstream investigation. So in particular, we would have gene expression data measured under two conditions. So in the very old days, maybe 20 years ago, people may use microarray to measure these gene expression levels in different samples. But later, since 2008, we have RNA-seq. So now this experiment is mostly done by RNA-seq. So what does the data mean? So typically condition one can be our wild type or default condition, and condition two can be our condition of interest. For example, we may have knocked out one gene, or we have performed some treatment to our sample, or it could be like a disease condition. So the question here is we are interested in differences. For every single gene I'm showing here as one column, I'm interested in knowing whether the gene has exhibited very different expression levels from condition one to condition two. So these genes may imply something related to the condition difference. For example, they could be biomarkers, or they could even be involved in the mechanisms driving the difference between the two conditions. So these genes will be screened out and we will study them further. So for this cartoon illustration, I just try to show that the left gene seemed to have some meaningful differences between the two conditions, but the right gene, not so much. So because RNA-seq, especially in 2008, right, even nowadays, is still not a cheap technology. So for an individual lab, if you want to study two conditions, you would usually not generate too many replicates from each condition. So 3 versus 3, 3 samples, 3 replicates under each condition is still the norm. So in this case, your comparison would only involve having 3 numbers to be compared with another 3 numbers between the two conditions. So how can we be so sure? So for this task, there are 2 popular software packages that were developed more than 10 years ago. They try to tackle the same problem: how to identify genes like the left gene, called differential express or DE genes, from the data. And they must account for the small sample sizes. Here, they are 3 and 3. And so to offer some statistical power, so this is a jargon, which means that we can actually learn meaningful signals by adding assumptions to really get around the small sample size. Because otherwise, with small sample size, we can say, oh, they could be some outliers, right? If they're outliers, they're rare occurrences, then maybe the meaningful difference is not here. So to overcome the small sample size, they use some assumptions, which is negative binomial distribution. So what is it? What is it? So if you have learned probability, negative binomial distribution is usually presented as an extension of Poisson distribution. Both are for counts. So essentially, this figure shows that if you have one negative binomial distribution and you draw a sample from the distribution, then the sample could be any of these counts, but at different frequencies. So in this case, if you get a count around 10, that is very likely. So if you repeat your sample many times and you summarize the frequency of each count value, you will get this distribution. So what they're trying to do here is that they assume the 3 counts, because RNA-seq data are reads, right? When we summarize the number of reads mapped to a gene, we get a count. The 3 counts here they are assumed to follow a negative binomial distribution. So that is assumption. But as you can see that this assumption is something we assume, but impossible to verify. Since you only have 3 values here, it's impossible to say whether they can be fitted well by such a distribution or not. Okay, so this is assumption, and they are popular software packages reflected by the number of citations and also by their very well-written tutorials. So I have actually encountered this problem in the collaboration project, which I found to be very interesting as a start. So essentially, my collaborator, whose credit I'll give at the end, a computational biology lab led by Dr. Wei Li at UC Irvine. So his former postdoc, Yu Mei, analyzed this data set previously published on Cell in 2017. So this data set contained 51 patients' blood samples pre-immunotherapy treatment and 58 patients on treatment anti-PD-1 therapy. So comparing these blood samples RNA-seq data, we may first run DE-seq2 and HR separately on the same data because both are popular software packages. But she found that the two methods at the same false discovery rate threshold. So that is a threshold, FDR is a threshold people try to use to calibrate the results to set the threshold for calling a gene as a DE gene or not. She found that the number of DE genes reported by DE-seq2 and HR are very different. And their overlap is quite small. So this triggered her question, why are the two popular methods identify very different DE genes on this dataset? So based on this question, and when we talked, I came up with this question about, can we try some permutation? The reason I came up with this question is because on this data set, the sample sizes are 51 and 58 instead of 3 and 3. So very different scenario from the case when DE-seq2 and HR were developed for. So they had to assume the negative binomial to gain some reasonable statistical power for finding DE genes. But this sample size is not in that range. So I said, okay, so now let's say we have 51 samples in condition 1, 58 samples in condition 2. Can we do some random permutation of the samples, but still keep the sizes as 51 and 58? But now the labels are no longer meaningful. So in this case, we can do thousands of rounds of permutation. Since we have a total of 109 samples, we just need to randomly choose 51 out of the total to create this fake condition 1 and the remaining as the fake condition 2. So for each permuted data set, we can then run DE-seq2 and HR again to see how many D genes are found. But to our surprise, this is what we get. So if you look at the bar, the height here is the average number of DE genes found from permuted data by DE-seq2 or by HR. And this error bar, half of its length, is the standard deviation of the number of DE genes found from permuted data because we had to do thousands of rounds of permutation. So this result is very shocking because it says that we can find even more DE genes from permitted data than from the actual data on many cases. So this cannot be true. And why this is the case? So my first thought is we should check the negative binomial assumption. Since now we have 51 count values for condition 1 and 58 count values for condition 2. We can just check whether the values seem to be agreeing with the negative binomial distribution. So we have enough data points to check the assumption. And to answer this question, why so many genes are identified from permitted data, we check the negative binomial assumption on the original data. And unfortunately, this figure, which is a technique we use in statistics, very useful technique called quantile plot to compare two distributions. This one, I'll just quickly say that it's based on the distribution. We fitted the negative binomial distribution reported by HR. And this is the theoretical distribution we should expect. So essentially, we're trying to compare what we get from the data to what we assume. And essentially, we see that if they agree well, if the negative binomial assumption agrees well, we should see the 51 data points lie perfectly on a straight line, on a diagonal line. But unfortunately, we do have some outliers here. The straight line fitting does not seem to hold well. For the other condition, we see the same thing, more severe deviation from the assumption from the straight line. So I would say that in this case, the negative binomial assumption does not seem to agree with the data. And why is that? The reason is when the methods were developed by DC to HR, they were developed, they assume negative binomial assumption. The negative binomial assumption has some theoretical ground on replicate data. For example, we can say that, okay, the 3 samples from condition 1 are biologically the same, except that we have different counts for genes in the 3 samples just due to the sampling process in sequencing. And that sampling can be modeled by Poisson. And beyond that, if we believe that the 3 samples, though biologically equivalent, may still have some subtle differences in the true expression level of a gene in every sample due to the biological stochasticity, we can add a layer of assuming these values to be different. And because of that, Poisson with this additional variation can be modeled by negative binomial. So negative binomial does have a reason or a justification for replicate data. But here, the 51 patients and the 58 patients should not be regarded as replicates because every human being is very different and we have the genetic variation. For all these reasons, the negative binomial assumption does not seem to hold well. So for people who analyze the data, another important question is, does it matter? If I know that the DE genes I found from my original data could also be found from permitted data, how would those false discoveries affect my conclusion? So here we just try to show that if you didn't do this sanity check or diagnostics we did, but you just take the D genes reported by DC2 and try to see which functional terms are enriched in those genes, you will actually be very happy because many of those functional terms are related to immune response. The same thing for the HRD genes. So the take-home message here is, if you just do this gene ontology enrichment analysis, you will be very happy to say, yes, I observe some meaningful differences between the pre-treatment patients and the on-treatment patients, and you can continue from there. But if you did the analysis we did, then maybe the data cannot give you a strong signal to support this conclusion. Maybe we should look into these outlier patient samples and see what they are, and to examine why they are different from other patients, instead of just saying, oh, we throw away these data points because they don't agree with my negative binomial, because every patient sample is very precious. One more thing I want to say for this work is that since the sample sizes are already much larger than 3 and 3, we have 51 and 58, we tried this Wilcoxon rank sum test, which is not an RNA-seq specific method, but a classical statistical method that does not assume the data to follow any pre-specified distribution. What it does, in other words, is trying to compare the 2 sets of data in terms of their ranks, whether you have the ranks in one set consistently higher than the ranks in the other set. By comparing the ranks, it is not relying on the assumption like negative binomial. So in other words, this one is more robust to model assumption. And by trying Wilcoxon, we can see that not to our pleasure, it does not find any D genes, which we don't like, but it's actually safe in the sense that it does not find any D genes from permitted data. So it is self-consistent on this data set. So I would say that for this data set, how do we do the data analysis while still getting some meaningful discoveries remains an open question. And this all boils down to whether these outlier values, how to use them or trust them. So I'll say that this work we published in 2022 surprisingly received a lot of attention, especially from the biomedical data analysis community, because many people never doubted that using a popular method may sometimes lead to problems and sanity check is necessary. So if you are interested in knowing more about this discussion happening 3 years ago, I was very happy to participate in the discussion because I found many questions people asked me on Twitter, then now X, were very insightful for me as a statistician, because even those very basic data analysis questions were still confusing. And I think we should do a better job in providing answers. So I want to acknowledge Yumei for leading this work. She's now a PI herself in Suzhou University in China. I'm my student, and then postdoc Xinzhou, who is now faculty at Oregon State, and my collaborator Wei at UCLA. So that's my story one. And my story two is about another common data analysis task, single-cell data visualization. Why do we need to visualize data? The problem here is single-cell data, every cell is measured with thousands of genes. So if you represent the cell mathematically, it's a 1,000-dimensional vector. But our human eyes can never see such a high-dimensional vector. We cannot see beyond 3D space. So what do? And biologists really like visualization. We rely on visualization to come up with new hypotheses from the data. So 2D visualization is popular, right? And we often show figures like this in our papers. So this is just a cartoon illustration showing that even if your original data is just 3D, not thousands of dimensions, but trying to show them on 2D, you must lose some information. Then the question is, what information do you want to preserve in 2D? What do you don't care so much? So for single cell data, people really want to see the cells of one type or of a known type as a cluster. So you try to visualize these cell types as clusters. And that is the reason why t-SNE and UMAP became popular 2D visualization methods. So both methods are called nonlinear dimension reduction, which means that unlike the more traditional principal component analysis, these two methods will perform some type of distortion on the data to encourage the neighboring cells in the high dimensional space to stay closer, to look like a cluster in the 2D space, and to push the data points or cells far away from each other in the high dimensional space to be even far away in this low dimensional 2D space. So we can clearly see the cell types. That's what they were developed for, and that's why they're popular. But people also realize that there's no free lunch. So both methods have their own hyperparameters that you can tune. And once you tune the parameters, your visualization may look different. So this is a commentary article from Nature Methods with this cautionary message saying that sometimes these methods take a second thought. So regarding t-SNE, which is an earlier method than UMAP, there is a very well-written blog post about how to use it effectively. So I took the screenshot from the blog post trying to show that the key hyperparameter of t-SNE called perplexity, which is related to how large you define the neighborhood size when you do the distortion. So if you set perplexity to be 2, you're assuming the neighborhoods are very small. When you set the perplexity to be large, you're assuming the neighborhoods are large. So under each perplexity, you will have a different visualization. And for this blog post, the original data is just 2D. So this is the true representation. And let's see how much t-SNE can distort. So you can see that when it's 2, very unrealistic. When it's 100, also not realistic because the two clusters are now mixed together. Only when your perplexity is in the middle range, you can see the two clusters. So this is one thing. Another thing is that when people visualize single cell data, sometimes they try to draw some conclusions by the distances of cell types in the 2D visualization. For example, they may say, okay, these two cell types are not so distinct by RNA-seq, by gene expression, but they're very distinct from the other two cell types. Then the question is, how much can we trust the visualization? To answer this question, we develop a method called scDEED. So the hope is to help people identify the 2D locations of cells that are not trustworthy. And our key idea is to examine the cell's neighbors before and after the embedding. So this method is already published, and we really welcome users to try our method to refine their visualization. So in short, I'll say that we try to define a reliability score for every cell by examining its neighboring cells before we do the t-SNE or UMAP embedding and after we get a 2D visualization. So we come up with two sets of neighbors and summarize their similarity into a reliability score. So this cell is something I try to show that the neighbors are very well agreeing with each other. And For this other cell, I try to show that the neighbors change a lot. So on the right, I'm plotting the pre-embedding neighbors before t-SNE UMAP, and we can see that on the 2D visualization, they do not seem to be neighbors anymore, and they're very different from the 2D neighbors. So in this case, the reliability score is poor. So one more thing we use statistics to help us is how to calibrate the reliability scores to say how high is it high enough so we can trust the location, or how low is too low, we cannot trust the location. So for this, we actually employ a very, I would say, simple but useful technique, permutation. So the idea is that for every gene, so every gene has a value in a cell, we randomly shuffle the gene's values across all cells. So by doing so independently for every gene, we are no longer getting the original cells, but we call them permuted cells. So for these permuted cells, they are actually exchangeable with each other. So every gene is preserved because its values are the same, but what's not preserved are the cell-cell relationships, because the permuted cells are no longer biological quantities and they do not preserve any meaningful relationships. So therefore, we can use this calculation, reliability score calculation on each permuted cell, and we can regard the resulting score as coming from a null distribution, because these scores do not reflect any meaningful cell neighborhood information. So we just need this permutation for once, and we can have the same number of permuted cells as original cells. For each permuted cell, we get a null value, then pooling together, we get a distribution. So only one round of permutation can be sufficient. We get a distribution. And on the distribution, we can define the reliability score to be high or to be low, corresponding to trustworthy locations or dubious locations. So regarding the application, I'll just show one example. So this is a science article showing the single-cell RNA-seq data from a species Hydra for the first time, and they're using the default t-SNE for visualization. So we can see many small clusters that may be hypothetical cell types. But when we apply our way to detect dubious embeddings in the original figure, we can see that some of the small clusters are now flagged as dubious, suggesting that maybe their locations shouldn't be trusted too much. Maybe they shouldn't be standalone embeddings, standalone clusters. So following this dubious embedding, what we can do next is to change the perplexity hyperparameter value to minimize the number of dubious embeddings. And this is the visualization we ended up getting. You can see that it's very different from the original one. We do not have so many small clusters. We actually unify some clusters into one. For example, for these 3 small clusters, even the original authors labeled them with the same label, but just used number 1 and 3 to mean that they look like separate clusters. But with our visualization, they're unified as one cluster, and this unification is also supported by the original gene expression data, because we do see that based on the original gene expression data, this orange stem cell cluster do look very different from those neuron ectoderm cells. But in the original visualization, they may seem to be close to each other. So I'll conclude my talk by saying that the technique I use in my 2 stories are permutation. So I use permutation as a way to add some statistical rigor to our data analysis. And permutation is listed as one of the most important statistical ideas in the past 50 years. So finally, I want to thank my collaborator, my former students, and also for scDEED, I collaborate with the statistician Lucy in Hong Kong and my student Christy. So thank you all for your attention.